Welcome to the Channelchek Takeaway series. This program is provided for informational purposes only, and none of its content should be interpreted as a recommendation or solicitation to buy or sell securities. Investment decisions must be made by you or with the support of your financial advisor. A complete disclosure statement follows each presentation. Onconova Therapeutics, NASDAQ ticker symbol ONTX, is a clinical stage biopharmaceutical company focused on discovering and developing novel products for patients with cancer. The company has proprietary targeted anti-cancer agents designed to disrupt specific cellular pathways that are important for cancer cell proliferation. Joining us today is Onconova President and CEO Stephen Fruchtman. Following a brief introduction to the company, Stephen will be joined by Noble Senior Research Analyst Robert LeBoyer for a Q&A. Take it away, Stephen.
So I will be making forward-looking statements as outlined on this slide. So who is Onconova Therapeutics? We are a clinical stage biotech company focusing on discovering in-house developed new medical entities for patients with refractory cancers. We have two proprietary targeted anti-cancer. Our lead medical entity is narazaciclib. I will take you through the mechanism of action. This is a CDK4/6 inhibitor, but what makes it unique is its safety profile, as well as the fact that it significantly targets other kinases that are important for both normal and cancer cell proliferation, as well as how cancer cells set up a new metastatic lesion. In addition, our second drug in development is rigosertib. Currently, rigosertib, which downregulates the overexpressed or mutated RAS pathway, or in fact, any protein pathway that has something called the RAS binding domain, such as PLK1, and is also an immune modulator that is being currently in small cell lung cancer and a rare disease called RDEB. And again, I will take you through both of these very interesting drugs in a little while. So here is where we are in the development. Narazaciclib is in a phase 1 study in both the US and China. And as in the first quarter of 23, we anticipate beginning a trial of narazaciclib plus estrogen blockade with letrozole in low-grade endometrial cancer. The reason we are conducting our single-agent narazaciclib trials in the US and in China is because CDK4/6 inhibitors, the ones that are FDA, are prescribed in two different schemes. One is a 3-week on, 1-week off scheme. The other is an everyday scheme. And when we started the phase 1 trial, we were not sure what the optimal scheme for narazaciclib will be, but based on the trial going on in China and the one in the US, we now have more information and recognize that due to an acceptable safety profile, narazaciclib can be administered every day. This is an advantage. Proliferate every day, and if you need a drug to be given and have a rest period of a week to permit something like the bone marrow to recover, it also permits proliferating cancer cells to recover. So based on the phase 1 we are conducting in the US, we already recognize the good news that narazaciclib can be given daily. And when we combine it with letrozole in the first quarter of 23, narazaciclib will be given daily. In addition, rigosertib is being investigated in KRAS mutated non-small cell lung cancer. Data was presented at ESMO. I will share that data with you shortly. In addition, as I mentioned, both oral and intravenous rigosertib as monotherapy is being studied in squamous cell carcinoma, complicating a rare disease called recessive dystrophic epidermolysis bullosa. When these patients develop squamous cell, there's no FDA that is effective for these patients. And we plan to begin a trial of oral rigosertib and pembro from Bristol Myers Squibb in checkpoint inhibitor refractory malignant melanoma. So let's talk more about narazaciclib. This is a pictorial of the CDK4/6 pathway. There are 3 FDA-approved drugs: palbociclib, ribociclib, and abemaciclib. These drugs target CDK4/6. When CDK4/6 is overexpressed in, let's say, breast cancer, the breast cancer cells, due to the overexpression of these proteins, proliferate, and thus the cancer cell has a proliferative advantage. It changes a resting cancer cell that's called the G1 phase. The stimulation by CDK4/6 causes it to leave the resting G1 phase and go into the synthetic phase. The 3 FDA-approved drugs target this pathway, as does narazaciclib, but we are different than these 3 approved drugs for reasons I will show you in a little while. We target other tyrosine kinases that are the putative causes breast cancer cells may be resistant to these drugs. These drugs currently are only approved in hormone receptor HER2 negative breast cancer, and all and men who get these drugs ultimately will be refractory, and we hope to change that refractory nature by targeting the putative proteins that cause the refractory breast cancer to become eminent. These are multi-billion-dollar franchises worldwide. The first CDK4/6 inhibitor was approved as palbociclib in 2015. Subsequently, the other 2 have also been approved. And as you can see, total worldwide, these drugs exceeded $6 billion and demonstrate that we have perhaps best-in-class CDK4/6 inhibitor with narazaciclib for reasons I will explain to you. One of the major reasons is I already mentioned, I'm going to show you the data. We target other proteins involved with oncogenesis, specifically CSF1. CSF1 is a protein that permits cancer cells, specifically breast cancer cells, immune surveillance. As you all know, the host immune system is very important in attacking cancer cells, but evading that host immune surveillance. And one of the ways of doing that is to use this protein CSF1. And I'll show you the data. ARC5, also known as NUAC1, in how cancer cells live in a hypoxic or low oxygen environment. So in a cancer cell, what kills women with breast cancer is the metastatic potential of the cancer. If it remained in the breast, it would not be lethal. But these cells tend to metastasize to the brain, the lung, the liver, the skin. And that is the reason why the patients ultimately have their demise due to metastatic breast cancer. But how does a cell that leaves the breast, goes to the liver, let's say, and set up a new nodule of breast cancer in the liver? Because at first, there's minimal, if any, blood supply to that new cancer. So the cancer has to wait for a new blood supply to be created by the cancer. But how does it exist without blood, without oxygen? So it has the ability to exist for a while in a hypoxic environment. And scientists have shown one of the ways it does that is through ARC5. Again, we target ARC5 very potently to prevent that cancer from living in a hypoxic environment. Another important aspect of narazaciclib many of the women die of metastatic disease to the brain. We've already conducted non-human primate studies. A non-human primate or a monkey is the best model to study the human blood-brain barrier that prevents drugs from penetrating into our brains. And in the non-human primate model, narazaciclib does get into the monkey's brain. Again, this will have to be shown as well in the human model. So here's the data I was referring to. This is the IC50 values. IC50. How much of a drug does it take to inhibit the activity? 50. These are head-to-head comparisons conducted under Onconova, comparing narazaciclib to the 3 FDA-approved drugs, palbo, ribo, and abemaciclib. If you look at just CDK4 and 6, the IC50s of narazaciclib very similar to palbo, ribo, and abemaciclib. But let's go to look lower down in this table. Let's look at CSF1R, the third row from the bottom, CSF1R. Look how potently narazaciclib targets CSF1R, the protein involved with immune surveillance. Only 0.7 nanomolar of the drug is required to inhibit 50% of that protein, when more than 10,000 nanomolars of the FDA-approved drugs are required. ARC5, again, look how much more potently narazaciclib targets this very important protein involved both in hypoxia and some believe in how cells travel and metastasize in the body. So based on the fact that we target other proteins involved in cancer more potently, we hope that this will be shown in the human model as well. The other thing that differentiates narazaciclib from the FDA-approved drugs is, I already mentioned, these drugs, 2 of the 3, have to be. The 1 week off is designed to permit the bone marrow to recover. This is a direct comparison in the mouse model of narazaciclib against palbociclib. And you can see palbociclib causes a 50% decrease in the white count of the mice. Narazaciclib, because it targets 6 as well, also lowers the white count of the mice. So we know we're hitting that target, but it does so in a statistically significant less manner than palbociclib. And most importantly, in our phase 1 study to date, we have not seen important neutropenia. We see a little bit of neutropenia, but not enough that the drug has to be stopped. So thus, the drug can be given in a daily fashion. This is a head-to-head study of narazaciclib against palbociclib in 3 different cancer cell models: breast, ovarian, and prostate. You can see when the cancer cells are exposed to palbociclib, there is some reduction in proliferation, mostly in breast, less so in ovarian, and less so in prostate. But look at how effective narazaciclib is in preventing the proliferation of all 3 of these cancer cell lines. And because these drugs are only approved in breast cancer, you can see how narazaciclib much more potently inhibits breast cancer cell proliferation in the left-hand cartoon compared to palbociclib. So that is the background of narazaciclib, but let's talk about pursue. So I already mentioned we will be studying narazaciclib in the most common cancer of the female reproductive organ, and that is endometrial cancer. So why are we doing it? These have already been conducted with the 3 FDA-approved drugs, palbo, abi, and ribociclib. Some of these have been randomized phase 2 trials. Others have been single-arm trials by itself, only a single arm of the experimental drug. But in randomized trials that have already been conducted, small numbers of patients. The first one was palbo plus letrozole, 36 patients versus placebo plus letrozole, the combination of palbo and letrozole had a profound improvement in the PFS compared to the standard of care, which is letrozole alone. Again, Abi and Ribo also have improvements in progression-free survival. We believe, based on the data I already showed you, the preclinical data, and this clinical data that was produced by the other CDK4/6 inhibitors, that we have a very most direct pathway to getting an approval in endometrial cancer with narazaciclib. The phase 1 study will begin any week now, hopefully sometime in the first quarter of 23. And hopefully by the end of the year, we will see evidence of efficacy in refractory endometrial cancer. We plan this to be done as a multi-phase trial. This is a little bit of a summary of the proposed trial that will be 3. So now I'd like to transition to rigosertib. We have much more clinical data in rigosertib. We're very excited about the preclinical data and the clinical data that's already been developed with the narazaciclib, but we have much more data with rigosertib, which I plan to share with you. So this is a cartoon of the RAS pathway. You may know that RAS is the most commonly mutated, and RAS sits in the cell membrane, and RAS, when it's turned on, and if RAS is mutated, as it is in a number of cancers, including non-small cell lung cancer, when RAS is mutated, it's always turned on. The area of this cartoon to the lower right-hand corner, when RAS is turned on, it causes the production of a protein called ERK, E-R-K, ERK. And when ERK is phosphorylated, cancer cells proliferate. So rigosertib inserts itself into the effector protein of RAS. So you see RAS, R-A-S, and then the next protein is RAF, R-A-F. And RAF is the effector protein that interacts with RAS and gets this cascade started. Rigosertib has the ability to insert itself into something called the RAS binding domain that RAF, R-A-F, has, and therefore downregulates this overexpressed pathway. In addition, other proteins, such as PLK1, polar-like kinase 1, also have RAS binding domains, and I will show you data with a disease driven by PLK1, where rigosertib again has very encouraging clinical results. You may know that Amgen and Mirati, for the first time, have FDA-approved drugs that target RAS, but their approved drugs only targets a specific mutation of RAS, KRAS G12C, because their drugs target RAS directly. Our drug, rigosertib, does not target RAS directly, and targets the effector protein RAF. Therefore, it does not matter. Mutation of KRAS may be in non-small cell lung cancer. Our hypothesis is rigosertib should be effective against any KRAS mutation, and now we have data to prove that hypothesis I will be sharing with you. So this is some preclinical studies we did with rigosertib that led to the non-small cell lung cancer trial. These are mice which have been inserted a human KRAS mutated non-small cell lung cancer in a xenograft model. This particular patient had KRAS mutation of G12D for dog, so neither the Amgen or Mirati drugs would have efficacy because they only target G12C for cat. So you can see tumor, when placebo is given, in the blue line, the tumor grows. When single agent rigosertib is administered to the mice in the orange line, you can see tumor cell proliferation is diminished. And then these human tumors are biopsied. In the placebo-treated mice, the upper photomicrographs in 5 mice, there's a lot of brown material. That's staining for pERK, the actual protein that causes the cancer to proliferate. Look what rigosertib does in the lower photomicrographs. Rigosertib prevents ERK production, and we believe that's one of the mechanisms for preventing the G12D mutated human lung cancer to proliferate in the mice. The other way rigosertib works is RAS, when mutated, cancer and ability to evade the immune system. It shuts down, in a way, T cells, host T cells, from recognizing that host cancer. In this case, what is being studied is malignant melanoma. But then rigosertib is put into the system, and it changes the cold melanoma cancer into what's called a hot cancer, that now malignant melanoma is actually attracting T cells into the tumor microenvironment. And therefore, if you introduce an immuno-oncology drug, T cells to help attack the underlying tumor. So rigosertib, through its ability to cause the cancer cells to express neoantigens, in this particular case, as published in Molecular Cell, CD40 is the antigen. This new antigen attracts the host T cells. And this is now a colorectal cancer, showing you the synergy based on the data I just showed you of when rigosertib is combined with a PD-1 inhibitor in a colorectal cancer model. So you see, when placebo is given to the mice, the tumors grow. Monotherapy with either rigosertib in the orange line or a PD-1 inhibitor in the purple line, both are efficacious in preventing tumor cell proliferation. But look at the combination of rigosertib and the PD-1 inhibitor in the green line. For the first time, not only are you getting tumor growth control, you're actually getting tumor cell death, because the green line is going down. For the first time, the tumor is dying, and you can see the synergy when rigosertib is given with a PD-1 inhibitor. Based on data such as that, we started a clinical trial in KRAS mutated non-small cell lung cancer. This trial was presented at the most recent ESMO meeting in September in Paris. You could see this is a report of 19 patients at the time that were entered onto this study. The type of KRAS mutation the patients had is listed here. As you can see, the most common mutation in this cohort of patients was actually G12V for Victor. So there are many different KRAS mutations, not just G12C, that the Amgen and Mirati drugs, miraculously, it's a fabulous advance that those drugs target G12C. But our hypothesis is that we target any mutation, including G12C, and I'll show you data, the clinical data, in a little while. These are very highly refractory patients. 80% of these patients received either 2 or more previous lines of therapy. All the patients failed Keytruda from Merck, and Keytruda is the standard of care. Keytruda plus chemotherapy. Highly refractory patients, 80% have failed 2 or more lines of therapy, and they have no other therapeutic options. This is a dose escalation trial with gradual increases of oral rigosertib, starting at 280 milligrams twice a day, and all the patients get full dose nivolumab as well, kindly given to the investigator by BMS. Here is the crucial efficacy data. So we have, at the time of this presentation, there were 13 patients who received rigosertib above the minimally effective clinical dose. 4 of 13 had disease control. 1 of the patients actually went into a complete response. So all sites of lung cancer in the lung disappeared, as well as sites of metastatic disease. 2 patients had a partial response, and 1 patient had stable disease. In each one of these horizontal bars, you could see which KRAS mutation the patients had. The patient number 8, who entered a complete response, had a G12V for Victor mutation. There's actually one patient who did have a G12C mutation. That's patient number 2, and also had a second mutation called STK11. Patients with STK11 have a very poor prognosis. The other partial response, patient number 14, had a KRAS Q61H mutation, and again, also had a STK11 mutation. You could imagine these refractory patients have the most aggressive mutation. So we achieved in 31% of patients who received above the clinically effective dose of rigosertib that we already knew had responses, again, 1 CR, 2 PRs, or 1 patient with stable disease. So these are very encouraging data for KRAS mutated non-small cell lung cancer. The study remains open, and we expect perhaps by the middle of 2023 to be closed. And again, we will update the data at a major medical meeting. Let's shift for a second to another disease now called RDEB, recessive dystrophic epidermolysis bullosa. This is a terribly fatal disease. These patients are born lacking a gene that makes a protein called collagen 7. In our bodies, we have a variety of collagens depending on what area of our bodies we're talking about. The skin has collagen 7. Collagen 7 binds our skin cells to the underlying dermis. If you don't have collagen 7, you slough your skin as though you were a burn patient with no skin. You could imagine many of these patients die in pediatric age groups of infection. The other major cause of death for these patients is the development of squamous cell carcinoma, which leaves the skin, may leave the skin, and metastasizes. These cancers are driven by a protein called PLK1, polo kinase 1, which has a RAS binding domain, and therefore rigosertib was studied in these patients. They are ultra rare, only 100 patients in the US per year. This data was presented at a dermatology meeting. The first patient is demonstrated here. You can see the left hand and the right elbow. So the underlying skin is abnormal. It looks white here, white areas of redness. But then in the middle of the picture is this large ulceration. On biopsy, that's squamous cell, and in this patient, the squamous cell metastasized. You can see in the lower photomicrograph how the large ulcerations, the squamous cell actually disappeared. All sites of metastatic squamous cell disappeared as well. And this patient remains on rigosertib now for 2 years and remains in complete response. More recently, we've treated our second patient. These patients are hard to find. And we continue to look for patients, and we anticipate that we will update this very interesting data with more patients at an upcoming major dermatological meeting. We are very encouraged by this, as is something called the Deborah Foundation, a foundation involved with patients who have RDEB, who've also provided non-dilutive funding for the continuation of this study to the investigator. So that is narazaciclib and rigosertib presentation. I hope you found those data very intriguing. We were very excited at J.P.Morgan to present this data. These data will be updated at major medical meetings, but we have some very important milestones coming up. We will initiate the narazaciclib endometrial cancer, as I mentioned, in the first quarter of 2023, the combination in women refractory with endometrial cancer. We will get narazaciclib plus estrogen blockade. Rigosertib, the non-small cell lung cancer trial, we anticipate will be finalized sometime first half of 23. And we will continue to study rigosertib in RDEB squamous cell as we find these individuals worldwide. Again, we anticipate updating the data at an upcoming major dermatological meeting. And with narazaciclib, based on our very intriguing IC50, we anticipate opening other trials in 2023, specifically for women, probably later in 23, once we have the recommended phase 2 dose of narazaciclib established more clearly. Women who are resistant, who have failed the FDA-approved CDK4/6 inhibitors, we anticipate opening up a refractory breast cancer trial looking at the role of narazaciclib because have generated as well. I thank you for your attention, and I'd be happy to take any questions you may have.
So one of the questions that I had was that in the phase 1 trials in the US and Canada, you're using several tumor types. What other tumor types is the CDK4/6 pathway active in? And what other indications besides breast cancer could narazaciclib be tested in?
Current phase 1 trial is an all-comer trial, all end-stage cancers, just to help us get as rapidly as possible to the recommended phase 2 dose. These drugs are only approved in hormone receptor positive, HER2-negative breast cancer. But there is preclinical data and, in addition, clinical data that CDK4/6 inhibitors are efficacious in endometrial cancer, but they're not approved in endometrial cancer. There is some off-label use. So our strategy, because of the preclinical data in endometrial, the fact that endometrial, like breast, is a hormonally driven tumor, driven by CDK4/6, our goal is to begin the phase 1 combination trial, specifically in endometrial, first quarter 23, have efficacy data by the end of the year, and then open a registration strategy trial, probably early 24. In addition to endometrial, for reasons I explained, we believe, we hope, we have preclinical data that I shared with you, that we have efficacy in breast cancer cell lines that are refractory to palbociclib, as you mentioned, with a better safety profile, narazaciclib, compared to the approved CDK4/6 inhibitors. So another indication we're very interested in pursuing is women who have failed the FDA-approved CDK4/6 inhibitors, and will begin a trial at some point in 23, looking at women with refractory to CDK4/6 inhibitors with HR-positive, HER2-negative breast cancer. We also have data in other indications as well, and stay tuned. We have submitted abstracts to major medical meetings for both mantle cell lymphoma, actually. And if you get away from CDK4/6 for a second, there is data in multiple myeloma, for instance, that patients who overexpress ARC5, that narazaciclib potently targets. Patients with multiple myeloma overexpressing ARC5 have a very poor prognosis. So our goal is first to investigate endometrial, shortly thereafter breast, and this is a one drug that hopefully is multiple franchises within a single drug based on the mechanism of action, as well as a better safety profile.
Great. And question on rigosertib is one of the things you mentioned was that this was an inhibitor of polo-1-like kinase, and it's being tested in the rare disease where polo-like kinase 1 inhibition has shown a benefit for the patient. First question would be, can you pursue this as an orphan drug? And the second is, are there other indications where polo-like kinase 1 is indicated and inhibition could be tested either as monotherapy or in combination with other drugs?
So thank you. So we clearly have a very powerful signal in squamous cell complicating RDEB. I mean, the results of the kind of docs, the dermatologists, the dermatological oncologists who see these patients are very excited by the data in squamous cell complicating RDEB. But there are other more common squamous cells of man, specifically squamous cell of the lung, squamous cell of the skin, not complicating RDEB, de novo squamous cell of the skin, squamous cell of the cervix. And we are looking into, so because there's no drug approved that targets PLK1, the pathology labs don't routinely check for PLK1 status. But now, as part of an investigational approach, we are looking at using AI and reaching out to pathology labs that we work with that have specimens to determine what is the incidence PLK1 overexpression in squamous cell, by the way, head and neck as well. And so there's a possibility, in fact, to have a histological agnostic study, meaning it doesn't matter where the squamous cell is, that if you overexpress PLK1 and if you overexpress PLK1, there's a possibility that you will be responding to Rigo, and that work is undergoing as we speak, both working with an AI company looking at the PLK1 pathway.
Very interesting. And like you said, if there's no treatment for PLK1 inhibition, they don't look for it. So in the presence,
we hope to have the first drug approved in squamous cell driven by PLK1, but clearly we have to do the work. But it's not the standard of care to check, and that may change. That may revolutionize. Yep.
Yes.
That's our goal.
That's commonly seen as to a problem that is known to exist, but can't be done. Nothing can be done about it.
Hit it on the head. It's not unusual that you could learn so much from one patient. A single patient with RDEB squamous demonstrated a remarkable response, a CR for 2 years in a disease that had no therapeutic options. Now we take that approach and expanded the studies into other squamous cells as well.
Great. Okay, well, thank you very much, Dr. Fruchtman. This has been a really interesting presentation. Just remind the audience that the slides are on the website if you'd like to look at them in the full set. And we look forward to following the developments and seeing these milestones in the coming year. Thank you very much.
Robert, thank you. And thank you to your investors who follow you and follow Noble, and hopefully are now aware of the very exciting research we are conducting at Onconova to help patients. Thank you.
With more than 6,000 small and micro-cap companies listed, if you're looking for the next Apple, then Channelchek truly is the orchard. It's an investor community where you can access independent Wall Street research, advanced market data, balanced news, corporate videos, and podcasts. You can even register to attend in-person Meet the Management Series lunches, and to be part of the preeminent annual Small and Micro-Cap Investor Conference, NobleCon. And here's the best news. Since Channelchek is sponsored by the featured companies, premium membership requires no investment. It's absolutely free. So check it out. Log in today. Find out why Channelchek is North America's leading source of research on the companies that just may change the world.